You're attending a webinar by OCS Life Sciences on the topic of SAS Visual Analytics for Life Sciences. Data visualization is a subject of increasing interest within the life sciences industry, and SAS offers Visual Analytics, or VA in short, which is a cloud and web-based solution that provides an easy interface to a large number of interactive data visualizations. This VA for Life Sciences demo has been developed by my colleague Femke Seidsma, who will take the word in a few minutes. But let's look at a few housekeeping notes first. You're attending a webinar, and that means that you can hear us, but we can't hear you. This ensures that all the participants can clearly understand our speaker without any disturbances. If you have any questions, you can write them in the chat box, which you'll find in the control panel on the right side of your screen, and Femke will answer all your questions at the end of the session. My name is Jules van der Zalm. I work for OCS Life Sciences as a manager, and I'm responsible for our project delivery. Next to me is my colleague Femke Seidsma, who is our data visualization specialist, who designs and develops in SAS Visual Analytics for our clients, and who has developed the SAS VA demo for life sciences. I will now give the word to Femke.
Good morning, everybody. Thank you, Jules, for your introduction. Yes, my name is Femke Seidsma, and I have created the demo that I will show in a few minutes. But first, I'll give a very short introduction of what you're going to see today. So I will demo SAS Visual Analytics in life science, and I think that's important because, for example, while running a trial, it is difficult to track progress or laboratory parameters while running the trial, and visualization can help by easily finding, for example, data errors, or just easily getting an overview of what's going on in your trial. So the demo of today consists of two parts, actually. In which the first part visualizes the recruitment of a study, and the second part visualizes clinical trial data. And then we'll be visualizing several domains, for example, just some demographics, some adverse events, and laboratory data. And the second part of the clinical trial data is subject specific, so really monitoring a specific subject. After demoing these several dashboards, I will also show shortly how to create a simple dashboard in VA, so you also have an impression how easy it is to create these dashboards. So now I will switch to SAS VA, which is a web-based program, basically, and this is the home page that you access when you access VA. At the top, you see several tiles: the report designer, the report viewer, the custom graph builder, data explorer, and data preparation. So the demo of today I've created in the report designer. And I will show it in the report viewer. So at the end of this demo, I will go back to the designer to show how I've created a dashboard. And the other parts of VA is unfortunately too much to show during this demo, but it offers more possibilities than I can show today, unfortunately. So I will go to the demo. And it has to load for a little bit, but that gives me some time to explain the first part of this demo. So the first part is the recruitment, and it visualizes recruitment in several studies. We've created demo data that shows 7 studies and 23 study sites, which you can see in this first dashboard, actually mapped on a geographic map. So here at the top you see the 7 studies in this dashboard, And on the left, you see all study sites. And then on the map, you can see where all the study sites are located. If you click on one of the studies, for example, Julius, the map zooms in on the study sites that are included in this study. And on the left side, you can also click on one specific study site, and it shows you the location of where this study site is. But just knowing where a study site is does not tell you that much about recruitment. So therefore, I created the recruitment progress, which is also a map, but this time it's animated. Here on the top, you see again 7 studies. Right now, Julius is selected, and the blue dots show where all the study sites are. And on the left, you see which study sites are included. If you press play in this dashboard, you see that the bubbles are growing. The bubbles are growing while more people are included in this study. And then also the bubbles turn green when the target for this study is reached, or for the study site is reached. If you hover over the study site, you get more information. So you get the name of the hospital in this case. You see the total subjects is 40. The recruitment target was 35, and they have recruited 20 females and 20 males. Then to show how recruitment goes over time, here's a line plot with recruitment over time. So on the y-axis, you see the amount of subjects that are included in this study, Adama. And on the x-axis, you see the study days, or the recruitment days. Then you see a light green and a light blue line, which represent the recruitment targets for this study. And the dark green and the dark blue represent the recruitment of females for green and the recruitment of males for in dark blue. You can see that in this study, both targets are not reached yet. And on the bottom of this page, you see also the 3 hospitals that are included, and you see that 2 recruitments of 2 study sites did not meet their target for females, and 2 study sites did not meet their target for males. If you go to another dashboard, you see that in this study, recruitment goes quite well. There's only a few sites that did not meet their target yet, but they are very close to what their target is. And you can also see in this table when the hospital started their recruitment and when they included their first patient. This is, of course, overall studies, and here you see the study sites again, but you could also look specifically at study sites, and that's what we can see over here. So right now, the study Eleonora was selected, and the London Bridge Hospital, and you see that basically the same line plot as before, but now it's study site specific. And you see again the 2 targets over here. And now you can see that for the London Bridge Hospital, the target for males was reached by day 75, and the target for females was not reached yet by the end of the recruitment period. And now we have, instead of study sites, we actually have subjects in the table below, in which you can find some, for example, in or exclusion criteria. And then the final thing for the recruitment is just some subject characteristics for the whole study, for the studies all studies specific and for study sites specific. So I've created butterfly charts over here with recruitment males on the left side and the recruitment of females on the right side. And then for different demographics or maybe in or exclusion criteria. So I have age here, I have BMI, I have whether people are smokers or not, and whether they consume alcohol or not. For now, I have nothing selected on the top and nothing selected on the left side. So if I select, for example, a specific study, I get the details about that study. If I select a specific hospital, I get information about who's recruited in this study or in this study site. If you hover over the bars again, you see that there were 3 males recruited in the age category of 36 to 50. So this was the first part about the recruitment. So now I'll move over to the clinical trial data. For this clinical trial data, we used sample data about a medicine called nicardipine, which was considered for the treatment of patients who had a particular type of stroke. In this data, we have 2 study groups, the NIC-15 group, which is represented in blue, and the placebo group, which is represented in green in all the dashboards that are coming. And we used visual analytics to visualize several domains that are good to, or just for general information like demographics or medical history, or really to monitor how the study goes and what happens in the study, which are more domains like adverse events or laboratory data. So for this first dashboard in the clinical trial, we see some general characteristics of the study. I've created 3 bar charts, one about sex, one about race, and one about age, in which you see just the distribution of several demographic groups between the 2 study groups. So on the y-axis, you see the frequency. So if you hover over the bars, you see that there were 281 females included in this study in the treatment group, the NIC-15 group. One of the nice things of VA is that it's really interactive. So if you click, for example, on this bar, you see that it brushes the other charts and tables in this dashboard. So you can actually see that there were 297 females in the placebo group, and you can see what the proportion is in each in the other demographic groups. And I always think it's nice to have a quick overview just about how many people do I have in my study, and how are they distributed over demographic groups, what's the mean age, which you can see here in the table, and see whether the groups are like evenly distributed. The same, so the brushing in the bar charts also actually happens in the table. So if you click on any bar in the table, the parts where, in this case, the male placebos are also highlighted in the cross table. And here at the right bottom, I have a small, basically listing of unique subjects ID, which also gets filtered by clicking on any bar. So now you see we have 29 other races, and in this chart you can see which unique subjects IDs these were, and you get some extra information about the age and the outcome of that patient eventually. And the other thing is that this table is used to link to the patient profile we see further in our demo. I will come back to that later. So now we'll go to medical history. Medical history, we've classified it in body system and organ classes, which you can see here. But not all disorders were coded, so we also have a part that is not coded. And we have created 2 buttons here at the top to actually zoom in on either the coded disorders or the not coded disorders. And on the left, you see, again, a similar cross table as we saw in the previous dashboard, just with the numbers of disorders that occur in this study. Because this is a lot of information, and it's maybe a little bit difficult to find out which number belongs to what disorder, we can zoom in with these 2 buttons at the top. And now you can see that, for example, hypertension has a frequency of 178 in the NIC 15 group. And again, when you click in the table, it brushes the bar chart. Then we go to adverse events, which is something you want to monitor throughout the study. And in this dashboard, you see a list table of, actually a list of, again, the body system or organ classes in which the disorders occur, and then 2 bar charts, one about causality and one about severity and intensity. Now, no body system or organ class is selected, so you see overall adverse events throughout the study and the distribution in causality and severity and intensity. If you click, for example, on a specific disorder, right now cardiac disorders, you see the frequency of cardiac disorders in the study. Cardiac disorders are occurring quite a lot because these patients had a stroke, and these disorders are very much related. Finally, you might also be interested in what the outcome of the adverse event is. So, for example, you want to see just the fatal or how many fatal adverse events there were, and you can easily select it with this drop-down list at the right top. And you see that in the NIC 15 group, for example, 15 people had a not related but fatal cardiac disorder adverse event. Then we go to the next one, the adverse event tree map, in which I visualize actually the same data as in the previous dashboard, but it's just a different visualization. Now you see on the right, again, the NIC 15 arm and all the adverse events that occur in the NIC 15 arm. And on the right, you see the placebo arm and all the adverse events that occur over there. The one extra information that is in this dashboard, which you can't see right now, is that adverse events are coded with MedRA coding. And this is the highest level of coding. So again, the body system or organ class. But if you're interested in more detail, you can actually drill down in this graph and see, for example, the high-level group term. And then you can even go a level lower and you see the high-level term. And again, if you select one condition or disorder, it gets brushed in the other tree map, the tree map of the placebo arm. And again, you can click again and again, and you're on the same level again. And you can easily go back to the overview by going back in the hierarchy. Again, causality and severity, you can select on causality, severity, or outcome of adverse events. For example, you might be interested in related adverse events and severe adverse events, and you see that they are only happening in the, or they only happened in the NIC-15 arm, which is as expected. Then we'll go to the adverse event line graph. Because you're not only interested in how often do adverse events occur, but also when do they occur, and do they occur at different times in the treatment group compared to the placebo group. So in this graph, you see again the NIC-15 group and the placebo group, and these curves represent the adverse events over time in the study. So I've plotted here day study -3 till study day 20, and the same for the placebo group. And you see that these curves are quite similar, although there occur a few more adverse events in the placebo group. Again, on the left side, you can select a disorder that you might be more interested in, for example, nervous system disorders, and you get to see the specific line of that disorder for each group. And this, you can also visualize this in a different way, which I've done in the next dashboard. So now this is actually, again, the same data, but a different kind of line plot. And now you see two groups in one line plot. Here you can zoom to specific days that you might be interested in. And you see already here that there's a really long tail of adverse events occurring for cardiac disorders. So there might be some data errors in this study. If you look, for example, at the total adverse events, you even see this a lot better. You see a really long tail before the actual peak of adverse events. And it goes both sides. So actually what's happening is that the first adverse event was already recorded 361 days before the study actually started. So this is obviously a data error, and you spot it directly when you visualize this data. So this is what I've created to monitor adverse events. And the other domain that I think is very important to monitor is the laboratory data that's done during the study. Again, you see on the left a list with all the laboratory tests that were done in this study, and right now glucose is selected. So in this bubble plot you see all the laboratory data for the whole study, for all study patients throughout the study. Every patient is one bubble. Again, the blue ones are the treatment group and the green one is the placebo group. And here you see the units, millimole per liter for glucose, and the results on the y-axis, and on the x-axis you see the day of specimen collection. So these patients had many laboratory tests done, and when they were out of the reference range, the bubbles turn red. So if I select another laboratory test, the dots change, and well, in this case, the unit stays the same, but you see a different distribution of laboratory data. One other thing you of course are, or probably are interested in, is specific patients or subjects that are included in this study, or that have specific out-of-range results. So you can see that this subject has a result lower than the reference, actually of 1.76, and the lower limit was 1.75. Maybe you're interested to look up more information about this specific patient, which you can actually do with the little table again on the bottom of left side. So you can type in the subject that you were interested in, and here you see the results for that specific patient for calcium. So you see this patient has 3 measurements of calcium. The one that was out of range we just selected, or we were just looking up, and you see that the other 2 were still within range. So maybe it's not too much out of range for this specific person. And again, this table we can also use to link to the subject specific dashboards, so which I will show later. Then finally, about the whole study characteristics, I've created an outcome at follow-up dashboard, which shows it's similar to the dashboards we already saw, a bar chart and a cross table with the frequency counts of subjects in each group, and also in this case the relative amount of patients that were in a specific, or that had a specific outcome. Again, if you click on one cell, it brushes the bar chart. So now I get to the subject-specific part of this demonstration. So right now we've summarized the data on a study level, and now we'll go to the subject-specific dashboards. And this first dashboard shows an overview of one subject throughout the study. So here you see which subject is selected, and here you, on the left, you see the different domains that are important for a subject to track or to get information on. So the first one is the adverse events. You can see the duration of an adverse event, and you can see the value or the severity of the adverse events. The dark orange shows that it's a severe adverse event. The lighter orange is a moderate severe event, and the light orange is a mild severe event. The same for a concomitant medication. You see the duration of a medication. In this case, the antibiotics were given for several days, and the analgesics also, and the laboratory data, ECG measurements and vital signs were regularly measured during the study. Oh, I'm sorry, I forgot to tell, of course, on the x-axis are again the study days. So you see that on the first, or 1 day before the start of the study, lab was tested, and again the color coding says something about whether the test result was within a range or was, in this case, below reference range. The bottom bar at the top you can use to zoom in to get a clearer view of what's actually happening or on which days measurements did occur. And at the bottom you can actually go directly to a listing of these patients for the several domains. And of course, for example, in lab, you can again color code what's going on. When is the test result within lab reference range, and when is the test result normal? And as promised before, it is possible to link to this dashboard directly from demographics, from the little table, or from the laboratory data from, again, this little table on the left. So that means this person is still selected, the one that I looked up, because the test result was out of range. So if I double click on this, I go directly to this patient. You see this changing to this patient's profile, and you have all the information about this subject in one dashboard. And the same you can do from the little table at the demographics. So it's really easy to link between dashboards using a small table, and you can easily look up specific subjects. Of course, the lab results, we saw the study lab results before in the bubble plot, but subject lab results are also important to look at. Again, on the left, all the different laboratory tests, and on the top, the subjects that's selected. You could select other subjects easily. And here you see a graph in which the green bars represent the reference range, and the dots actually represent the test result. And when the test result's out of the reference range, it's red. So in this case, it's below reference range. And in this case, it's above reference range, so they are both red. But if it's within reference range, it's green. And at the bottom of the dashboard, again, which test are we looking at? Carbon dioxide, and what are the results? And some more information, even which date this test was done. Then we have, we are often looking at change from baseline in clinical trials. Again, a specific subject is selected. Now I've selected carbon dioxide, and here you see the change from baseline. So here's again the change, or now the y-axis is the change from baseline. The x-axis is the study day of specimen collection. And we see that compared to baseline, there was a drop for the first measure, or the second measurement actually, of -17, which you can also see here. So this was the baseline test result, and it dropped to 91.31, which is a drop from 17.39. And this is outside of the normal range, so the bar turns red. And here as well, and the others are still within reference ranges, they are still green. Then I have one final demonstration of all kinds of other graphs that are possible in VA to create, but yeah, it's just a snapshot of different graphs. So we see, for example, a word cloud of the adverse events in placebo, and where larger letters represent a larger frequency of this disorder. We have a heat map of causality and severity, in which you see that the most adverse events were not related and actually mild. You can create box plots for looking at distributions of, for example, lab tests. So right now we have selected hematocrit. We see NIC-15 group and the placebo group and several study days, and you can see how this specific laboratory test was distributed, and you get, if you hover over it in the data tip, a lot of information, for example, the average and the median. So it can give you a really quick overview of how the lab data looks like. And finally, we have created a waterfall chart from also from change from baseline. Again, when it's red, it's out of range, and when it's green, it's still within the reference range. So these are just a few examples of other charts that you could create in VA. So as promised, I will finally show shortly how it works, or how you design a dashboard in VA. So we'll go to the report designer for that. And I will just create a simple chart just to show how dashboards are created and how easy it works. So if now we're instead of the viewer where we were before, when I showed the demo, we are in the designer, and this is the designer part of VA. Here on the left you see 3 tabs, and here on the right you see a couple of tabs that I will show. But first, of course, we have to select a data source. So for this, I will just recreate some demographic graph. So I've used DM data for that. And now you see here on the left all the variables that are included in this data set. Then we have the objects, and as you can see, there's lots of objects that can be used within SAS VA, and I've only showed a couple, but there's a lot more. And there's also the possibility of customizing graphs when they're not, when you have a little bit different data. It can be good to use a standard graph, but just adjust, for example, some horizontal or vertical axis. For now, I'll create a very simple bar chart, and I can drag and drop it to the field in the middle, and then you see directly on the right that you can select roles here. Oh, I have to move this, and I hope I can do it like this. Oh. Okay, I'll try to work around it. So we have to fill in the variables that we are interested in. I want to recreate the bar chart about race, and I can drop race over here as a category. You could also select it here, but I can't get to the arrow right now, so I just do it like this, and that works as well. SAS VA automatically counts and creates a measure that's frequency, so you don't have to do anything to create or calculate frequencies over here, but we are interested in the different arms in our study. So we also want to add description of planned arm. And then I can assign how I want to use this variable in my graph. So here I already have race, and I want to keep that, but I want to group by my different arms. So now it's actually almost the same as the charts that I used in my demo, but will show some styling things you can do in this designer part. For example, I've applied in my demo sometimes a data skin, which I think looks nice, but you can try different or apply different data skins. You can adjust fonts or enlarge or the size of the text very easily. And finally, I have add a filter to the data in my demonstration. So I actually filtered out the screen failures, and now you see that they are still there, which is really easy. You can easily add a filter to a dashboard by clicking on filters, selecting the variable that you want to filter on, and just uncheck the screen failure. So now it's actually the same bar chart as I had in my demonstration. So that's a really quick overview of how the dashboard or the report designer works and how to create a dashboard. With that, I actually come to the end of my visual analytics in life science demonstration. And so I will have a very short summary because we're, I think, quite good on time still, but I want to keep it like that. And I want to give you some opportunity to ask questions as well. So in short, I just showed several dashboards visualizing recruitment in study and several domains in a clinical study. And at the end, I showed how you design a dashboard in visual analytics. Some benefits of visualizing data are that it, for example, can improve efficiency and effectiveness of data review processes, as you can set up your dashboards in such a way that data gets refreshed automatically, which I couldn't show right now, but you can set up the server that it auto-loads your data to SAS VA, and so it refreshes your data when there's new data added. So you're always up to date with your dashboards, actually. Another benefit of using visualization for clinical trial data is that you can set up alerts. For example, when a laboratory test is out of range, you can set up an alert that you get an email just showing that there was a laboratory test that was out of range, so you can check how that patient or subject is doing, or whether it's a data error. So it works really quick in checking your data and monitoring your data. Finally, the linking between the dashboards, as I showed, for example, looking up a specific subject and going with a few mouse clicks to that subject details really helps in both summarizing the whole study but also checking specific subjects and going to details or listings with just a few mouse clicks. So I actually hope you enjoyed this demonstration of SAS Visual Analytics, and I want to thank you for your attention. I'm happy to answer questions, so I will give the word back to Jules, who collected your questions from the chat box. Thank you.
Thank you, Femke. That was really good. We've received a few questions, which we'll get to in a minute. Let me just remind you that if you want to ask a question, the control panel of the webinar software at the right side of your screen has a questions box where you can type out your questions. We can't hear you, so you need to type them down there, and we will answer them. All right, question number one, Femke. If you use SDTM data, are you at an advantage?
Thank you for your question. I think you are a little bit or you have a little bit of an advantage. In my demo, I used SDTM data, and it works really nicely in the sense that it is standardized, and so between data sets, variable names are the same, and which makes the linking between the dashboards easier. So I think you would have a little bit of an advantage, but it would mostly be in the data preparations, which I didn't show actually in my demo. But actually, for that, you could use any standardized data. So if you have like a company standardized data, that would work as well. But also, if your data is not standardized, the data preparation might take a little bit longer, but still, SAS VA really doesn't mind whether your data is standardized or not.
All right, thank you. Next question. What is the single biggest business benefit you see in visual analytics?
Well, of course, I'm focused at life science. So for life science, I really think it's a great tool to really monitor your data and really have early data access. You don't have, as I said before, you don't have to already have standardized data. You could also import your raw data to SAS VA yeah, really look at how your data looks like instead of only or just only looking at it when your data is collected and processed for creating listings or things like that. You have really an early look at your data, and you can monitor your data really easily.
All right, thank you. Next question. How much time does it take to create a dashboard?
It depends a little bit on how your data looks like and your experience with VA. I think Visual Analytics has a quite steep learning curve, so which means that in the beginning it can be a little bit difficult to find out how your data should look like to create a nice graph in Visual Analytics. But as soon as you have a feeling for that or develop a feeling for how your data should look, as you saw, it really works quickly to create a dashboard. So I think, to be honest, to recreate dashboards that were in my demo, I've already prepared them once before. So depending on your data, it would take me a couple of hours to half a day to recreate dashboards that you just saw in my demonstration.
Thank you. Next one. I noticed that classifications like relationship to study product and age classification were ordered alphabetically. Can I also select a logical ordinal sorting?
Yes, you can. You can, in the designer, there's the option to make a custom sort. So you can sort it exactly the way you wanted it sorted. For example, the outcome at follow-up, I didn't order alphabetically, but I ordered them which was for me a logical order and which looked nicer in the graph. So yeah, it's easy to custom sort your data.
Great. If you have your own EDC platform, can you then link directly to that data, or do you have to import all your datasets manually first?
I'm not sure whether I understand what you mean. I actually don't know what an ADC platform is. Maybe my host, my colleague Jules can.
Sure. EDC is where the data is captured and where the data is eventually stored.
Oh, okay. Well, I'm not sure whether I'm answering your question, but the way I set up my data is that you have all my data set on an autoload server. So my data is on a server, and as soon as I update my data, I write it to, or I overwrite it to that server, and VA automatically uses this last data sets. So I think if you have the technical possibility of linking VA to your server or your data capturing system, yes, you could easily use that, and but that's a more technical part of VA that I'm not used to. You should ask like an administrator how to set that up properly, but it is possible to link your server to VA and get autoload or automated updates of your data.
Right, so I imagine as soon as VA can reach the location where the EDC data is stored, it can load it every time it's updated.
Yeah.
Without manual interaction.
Yes.
All right, great, thank you. Is it possible to hide the patient data related to the graphs in the case of blinded staff? And can you then hide only some data?
That's hard for me to visualize how that should look. I did create, I'm not sure that answers your question, but I did create graphs where the study groups were still blinded, so we couldn't create groups like I did in my study with the treatment group and the placebo group. So yeah, of course it's still possible to create graphs or plots using blinded data. Really hiding data for specific viewers, I'm not sure how that should work. I would have to look into whether that's possible and how that should work.
All right, thank you. It seems that we're getting quite a few questions, which we're of course very happy with. If it appears that we cannot make it within our assigned time slot, then we will get back to your questions outside of the webinar session, so don't worry. Next question then. In your presentation, you segregate data by domain and table. Is it possible to graph all the information present for a specific value present in different domains? Maybe you have to read it.
Let me see. For a specific value, I think you mean whether you can have like the adverse events and the concomitant medication and lab in one table, and for example one subject, and that is possible. But in my data I have separate data sets for each domain, but that's just a preference of how you like to work. But you can of course join these tables on subject ID and create one large table which includes all domains you're interested in. So yes, that's possible.
All right, thank you. Next question then. Is it easy to share reports, and how does it work?
Yes, you can easily share reports, and there are different ways of doing that. As said before, your administrator of SAS VA, which I'm not, can actually give rights to each person that can access VA. So within VA, you can design your report and share it with other VA users, and dependent on the rights of that user, he or she can only see the report in the viewer, or he or she can actually make adjustments in the designer, or you can co-create a dashboard together. So that's one way of sharing it. When you're done with your report, you can send emails to the other VA users to let them view or work further on the report. And the another option is creating a PDF of your report, which goes really easy. You can do it either in the designer or in the viewer, and SAS VA automatically creates a PDF of your whole report or parts of your report. Of course, you lose a little bit of the interaction. You can only, for example, show one patient profile at a time. But if a person is interested in one specific subject, you could easily print out the tables and the patient profile for that subject and distribute it as a PDF. And in that PDF, you see first like a snapshot of, for example, a table, because the table does not fit on a just a single page. But then there is a link to the bottom of the PDF, which shows the whole table with all the information for that subject. So that works actually really nicely. And the last thing you can do is also basically share your data. So if you are on a specific graph, you can transport your data from VA to, for example, Excel, and just have a snapshot of your data in an Excel file and share that with colleagues or whoever it needs.
All right, great, thank you. We are running out of time. We will make sure to gather all remaining questions and answer them individually. Also, if you have any further questions, don't hesitate to contact either Femke or myself. Our contact details are on the screen. All attendants will receive an automated email after the session that you could also reply to with any further questions or comments. We've recorded this session. We'll make it available on our YouTube channel. We'll send a link to you via email as soon as it's been published, and we'll also include it in our next newsletter. Thank you for attending our webinar, and we wish you all a very good day.